So first of all, I'm going to do a quick review here, just overview what nucleotides are. Nucleotides are basically made from three components: a nitrogenous base, a ribose sugar, pentose sugar, ribose sugar, and one or more phosphates. That's what the term nucleotides refer to. So ATP, GTP, UTP, CTP, what have you, they are all nucleotides. The diphosphates and the monophosphates are also nucleotides. Okay, I want you to kind of get your head around that nomenclature. Now, nucleotides are extremely important. First of all, when we talk about ATP, it's a major energy source, so we're gonna have to make it all the time. Right? So very important energy molecules. Very important structural components of coenzymes like NAD, FAD, coenzyme A. Very important in activating certain molecules. You remember UDP glucose. You need uridine diphosphate. That's a nucleotide coming to activate glucose to get into glycogen synthesis. Most importantly, nucleotides are needed to make nucleic acids, DNA and RNA. And both of these, for some of you who may not be taken or haven't taken a lot of molecular biology, these are the essential component molecules for cell division, protein synthesis. Okay? If you have neither or either one, cells cannot divide, you cannot make cellular proteins, and cells die. So nucleotides are the building blocks of nucleic acids, RNA, and DNA. That's very important. Fifth important thing about nucleotides is because of their importance, they have been very good targets, therapeutic targets. As I just mentioned to you, antimicrobial drugs target specific enzymes in nucleotide affecting nucleotide synthesis. Anti-cancer drugs do the same. Antiviral drugs do the same, target specific enzymes in nucleic acid and nucleotide synthesis affecting nucleic acid synthesis. So like I said, the concept is very simple. If you inhibit the synthesis of a single nucleotide, you can inhibit all of DNA or all of RNA because you need all four. Remember? In DNA synthesis, the codon needs all four nucleotides. You can't use only three. So if thymidine is missing, that's it. You're done with DNA synthesis. If thymidine triphosphate. So really very important concept. Okay, so there are two kinds of nitrogenous bases that make up these nucleotides, purines and pyrimidines. And the purines are adenine and guanine, and they're used in both DNA and RNA synthesis. There are three pyrimidines. Thymine and cytosine is used in DNA, and cytosine and uracil is used in RNA. So this is a summary slide of this. These are the purines used in both DNA and RNA. Only two, adenine and guanine. Three pyrimidines, thymine, cytosine in DNA, and cytosine, uracil in RNA. You don't need to know structure or anything, but please recognize that purines have two rings. That's helpful. And pyrimidines have one ring. There are a few other not so common nitrogenous bases called hypoxanthine and inosine monophosphate is the nucleotide derived from that nitrogenous base. Actually, this is a very important intermediate, inosine monophosphate, which contains hypoxanthine as the nitrogenous base, abbreviated IMP. This is actually considered the parent purine nucleotide. Okay? The first nucleotide, purine nucleotide made, and from it we can make AMP and GMP. There are a few other ones, xanthine monophosphate. These are just metabolites during degradation. There are some more nucleotides. We're not going to get into them. They're unusual bases. Containing unusual bases. Typically, these bases have, for example, dehydration, dihydrouracil, acetylation, methylation. They're typically found in tRNA and viral DNA. Okay, so you don't need to, but they're derivatives of the common nitrogenous bases. Now, if you take a nitrogenous base and add a sugar to it, a ribose or deoxyribose sugar, it becomes a nucleoside. Okay? So if you add a ribose to adenine, it becomes adenosine. Guanine becomes guanosine. Cytosine becomes cytidine. Thymine becomes thymidine. And uracil becomes uridine. So these are, once you see those, these contain both the nitrogenous base and the ribose or deoxyribose. The presence of the ribose creates the side, that's the sugar part, nucleoside. Okay? So here's the ribose. See it? Hydroxyl group. This is 1, this is 2. Deoxyribose is the hydroxyl at the 2 position is missing. So for RNA nucleotides, you need the ribose sugars. So this would be cytidine. You add cytosine here, be cytidine. So this is a nucleoside. And if you add adenosine to a deoxyribose, you form deoxyadenosine, and that's what you will find in DNA. Deoxyribonucleotides are needed in DNA synthesis. Finally, when you add the phosphate to the nucleoside, here's the nucleoside, and this base can be any of the nitrogenous bases, a purine or a pyrimidine, and you add a phosphate to it, the entire molecule becomes a nucleotide. 1 or more phosphate, up to 3 phosphates, to form nucleotide triphosphates, or 2 phosphates would be nucleotide diphosphate and nucleotide triphosphate. Okay? So that's what we usually need. When we want to make DNA, we need NTPs. When we need to make RNA, we need NTPs. Actually, for DNA, you need deoxy NTPs. That means the ribose is a deoxyribose. So in nomenclature, it's pretty easy. You start with the base. So if you have adenine added to a ribose, it becomes adenosine. If it has 3 phosphates, it's adenosine triphosphate, and that gives you ATP. If the ribose is deoxyribose, then you put that deoxy before adenosine, a prefix. and it is abbreviated as a small d as deoxy ATP. So what you need for DNA, for example, synthesis is deoxy ATP. What you need for RNA synthesis, you need ATP. And obviously the other nucleotides, triphosphates as well. Okay, so I'm going to get started. Just give me a couple of minutes and just to get you going on this one. And so basically, we're not going to cover a lot of details here, fortunately, because this is pretty complex pathway. But we're going to start with purine nucleotide synthesis. And we're going to mainly want you to concentrate on the rate-limiting enzyme and how it's regulated. And we're going to cover a few things regarding a couple of diseases, inherited diseases. And obviously, we're going to cover some drugs that affect purine and pyrimidine synthesis. Okay, so first of all, I want you to appreciate that the ribose needed for nucleotide biosynthesis, purine nucleotide, comes from H&P. One of the products of H&P pathway is ribose 5-phosphate. Very important. Okay. The second thing you want to know about nucleotides, you got to remember nucleotides contain nitrogenous bases. Those nitrogenous bases are derived typically from amino acids. So this is a very important slide. You need to know what are the contributor amino acids to that purine rings. Glycine, aspartate, and glutamine. 3 amino acids. Okay? You don't need to know the numbers or where they go in, but just know 3 amino acids needed for purine nucleotide synthesis. Glycine, glutamine, and aspartate. Also, importantly, I want you to pay attention to these guys. These carbons are coming from THF, tetrahydrofolate, what we just talked about as the active form of folic acid. So without tetrahydrofolates, purine nucleotides cannot be synthesized. That's how important THF is. Okay? So know the 3 amino acids, know that purine nucleotides require THF. So before you start purine nucleotide synthesis, here's the ribose from H&P. You need to activate it. The way you activate it is through an ATP. The enzyme is called Prpp synthase. Phosphoribosylpyrophosphate pyrophosphate is what is made. This is going to be the starting material, the activated ribose. Okay? The enzyme has another name, ribose phosphate pyrophosphokinase, but this is the common name, Prpp synthase. So this is what you're starting with. Okay? Prpp. This is the rate-limiting enzyme. I want to start with that, and then I'll stop, and then we'll continue on Friday. The first step of de novo purine synthesis is the rate-limiting step. You're going to take that Prpp and add to it part of an amino acid, nitrogen, from glutamine. So the enzyme is called glutamine phosphoribosyl pyrophosphate amido transferase. That's the rate-limiting enzyme. It's a mouthful of an enzyme, but it can be abbreviated glutamine Prpp amido transferase. So that's the beginning of the building of the two rings that make up the purine rings. The first enzyme, glutamine Prpp amido transferase, very important. You're starting out with Prpp, you need glutamine, both can be considered substrates, both activate this enzyme. They go up, they both can activate the enzyme. You always want to think that this enzyme is going to make all results in the synthesis of all purines. The first purine made will be IMP, inosine monophosphate, that I mentioned before. From it, you will make GMP and AMP. Okay? So, here's this enzyme. Here's the phosphoribosyl phosphate. You take the amine group from glutamine and start building that 2 rings of purines. Importantly, if you have too much IMP, GMP, or AMP, they can inhibit this enzyme. So they regulate their own synthesis that way. The activators of this enzymes are really both Prpp and glutamine. Okay? So this is the, I'll stop after this slide. I'm kind of on a roll here, but this is, I just want you to appreciate one thing. This is the only enzyme I'm asking you to learn. Okay? Look here. Look how many enzymes we're skipping. Okay? But the first purine made is this one, IMP. That's the first thing to pay attention to. That's considered the parent purine. From it, we can make AMP and GMP. Also importantly is to note that the synthesis of purines requires THF. One time here, another time there. 2 carbons, 2 formal groups coming from THF. And that really takes us to the strategy we talked about. So for microorganisms, you can inhibit the synthesis of purines by inhibiting the PABA, which actually inhibits folate synthesis. And dihydroteroate synthase for this one here. And for humans, we can use methotrexate to inhibit dihydrofolate reductase and inhibit the synthesis of THF, and that's chemotherapy drugs, and that's what we covered earlier. Okay, so 2 strategies there, both affecting eventually THF synthesis and therefore purine synthesis.